Transcribing recording is about to start. Hello, hi, how are you? My name is Brenda Amata, and this is
Sarah Kumi.
we are students from the Department of Biology at the University of Texas Arlington with Dr. Pia Jose. We are going to be speaking on the expansion of female diagnostic protocols in muscular dystrophy.
Based on our study, we propose altering diagnosis protocols for females with muscular weakness informed by a defective gene for dystrophin and adding affected female individuals confirmed through multiplex ligation-dependent probe amplification to the research population for new and current therapeutic strategies. Our proposed outcome is to decrease the age of diagnosis and determine better therapeutic strategies for varying progression of symptoms.
In muscular dystrophy, it occurs through the genetic mutation that happens in the body. There are 9 major forms of muscular dystrophy that affect all ages and sexes. So, with research, we have found that females get affected due to the sex-linked inheritance, and they maintain a functional genome that reduces the chances of MD or lessening the symptoms. According to Consumer Reports, most females who are carriers often undergo undertreated or misdiagnosed or completely have their symptoms ignored when affected. With new health innovations, we have come a long way with new diagnostic tools, and with early treatment and interventions, we can start to minimize severity.
Our key concepts include newborn screening through introducing blood and urine tests. DNA analysis can be completed to determine whether the female is a carrier or affected. For infants and toddlers, female carriers already exhibit unexplained symptoms and may be completely ignored. By using electron microscopy, MD can be identified by changes in the muscle fibers. Children 2 to 5. Some children may exhibit some symptoms indicating MD. Using repetitive stimulation to assess neuromuscular function, the extent of MD can be determined. Children's 6 through 12. Most symptoms are exhibited at this time. However, most females indicate mild symptoms. Using diagnostic imaging tools such as phosphorus magnetic resonance spectroscopy, one can determine the amount of energy within the muscle cells. For adolescents and above, immunofluorescence testing can be used to detect specific proteins such as dystrophin within the muscle fibers. Use of electromyography can help record muscle fiber and motor activity in carriers, which is very useful as it indicates unusual changes from the first test. For expectant mothers, although studies have shown improvement in testing safety during pregnancy, the risk is still high. With extreme caution and understanding of the risks, expectant mothers can undergo genetic testing by amniocentesis or chorionic villus sampling.
On our discussion, we have found that the most common forms of MD are Duchenne, which is DMD, or Becker, which is BMD, affecting females differently in part of the protein dystrophin, which is found in the X chromosome, which definitely undergoes the mutation. Females have 2 chromosomes, hence, most females are usually maintain a functional genome in itself, and some of them become carriers and some of them become manifesting carriers. So, with this, we have found that introducing new protocols to diagnose females throughout their lives, especially the carriers, gives early treatment and interventions. Research indicates that protocols placed in for people who have the BRCA1 and BRCA2 breast cancer genes have successfully lived fulfilled lives due to the early screening of our risk assessments. Most medical treatments may include ACE inhibitors or alternative, where with all the studies needed, we have become to determine that the prevalence for cardiac complications in females, carriers, and altering diagnostic protocols will aid in this goal.
Early screening of muscular dystrophy leads to lesser severity of symptoms incurred. This, in combination with the continuous screening in female carriers, ensures early detection of any symptoms and/or changes in muscular structures. Proposed guideline changes for diagnosis of DMD/BMD in women that retain some functionality of the dystrophin gene may allow for more successful and novel treatments and interventions, especially as earlier screening may decrease the diagnosis age of DMD/BMD. And diagnosis may occur before the symptoms emerge. Earlier screening may also introduce less aggressive treatments and interventions like physical therapy in lieu of medications and gene therapies.
And in acknowledgement, we would like to thank Dr. Jose for her support and guidance throughout this process, and our classmates for their contribution that inspired this project. Thank you so much. My name is Brenda, and this is
Sarah.
Thank you. Bye bye.